Hi there, my name is Luis. I'm a lecturer at the School of Applied Mathematics at the Getúlio Vargas Foundation in Rio. And today I'll tell you a little bit about MCMC for trees, in particular with applications to Bayesian inference. Right, so before I do that though, I would like to acknowledge my wonderful collaborators on this project. Andrew Rumble was my PhD supervisor at the University of Edinburgh. He's a professor of molecular evolution. Mark Suchard is a professor of biostatistics at UCLA, plus a bunch of things. And Guy is an associate professor at KU Leuven. Right, so what's the plan for today? I'll tell you what the problem is that we're interested in and why we're interested in that problem. And then I will tell you a little bit about the parameter space, so where the objects that we are interested in live. Then I will tell you about MCMC in this space, in particular adaptive Metropolis-Hastings. And then I'll tell you just a little bit about some results. We had some preliminary results, and then some perspectives, which is really the most important part of this presentation. Before I truly start, I should say that my main goal with this presentation is not necessarily to show you some really cool work that I've done, but rather is to motivate you to have a look at phylogenetics and MCMC in this space, because this is a very exciting new area of research. And by the end, I hope I'll have motivated you, whether you're a more mathematically minded person or a more practically minded one. I think there are opportunities of research for everyone, regardless of their sort of more quantitative or not inclinations. Right, so the first thing that I should tell you is what objects we're interested in, right? So what's the problem here? So what we have is we have populations, so we have biological populations, and the form, so the dynamics, the population dynamics these populations go through end up shaping the sorts of phylogenies that we obtain when we construct phylogenies from individuals sampled from these populations. So here in this slide, what I'm showing you is, or are, two populations. One which is growing, was not growing at all. It's stable through time. And then I sample some individuals, right, through a coalescence process. And this is the sort of tree that I obtain. So here, the yellow dots are unsampled ancestors. So these are people that existed, or individuals that existed, but we didn't sample. And the red dots here are individuals that we did sample. Now, the first thing to notice here is that we are sampling these guys through time. So this is what we would call a serially sampled data set. We are sampling these guys through time, and I hope you will appreciate that this induces some constraints on which phylogenies we can estimate. Because in whatever the phylogeny is, this guy needs to come before this guy. If time is growing in this direction, these guys need to come before this guy. So there's a temporal precedence constraint that needs to be satisfied. Right, but the other thing is, when the population is growing, say, exponentially, this will imprint this something on the tree. So this will leave a mark on the sort of tree that we are going to reconstruct from, let's say, molecular sequences from these individuals. So different population processes will lead to different phylogenies. Right, and we can make this relationship more formal. So we can treat this relationship formally through very simple, relatively simple mathematical models. So here, what I'm showing you is a model where the waiting time between coalescence events. So this is a coalescence event, this is another one, and this T4 is the waiting time between this coalescence event and this one. So the waiting times can be modeled through exponential distributions. So an exponential distribution with this rate. And this rate depends on the population size. So in particular, this is a model for a constant population, so a population whose size doesn't really change over time. And NE is a parameter that measures the effective population size, which is constant, obviously, over time in this model. So I can relate the population size with the lengths of the branches in the tree, so the lengths of the edges. So these trees are nothing but planar graphs with attached lengths. Right. And so we're interested in these things because we can collect loads and loads of molecular sequences, sometimes complete genomes, from viruses and bacteria, pathogens in general, and we can construct phylogenies, and then from these phylogenies we can make useful inferences about the population processes shaping the evolution of these pathogens. So here I'm showing you another iteration of the previous slide where different time dynamics lead to sort of different tree shapes. And here is an illustration of what you can do when each sequence comes from a particular location. So the pattern, the clustering pattern of these sequences in the tree will tell me something about the geographical dynamics of this, say, viral population. Right. So how do we represent this space of trees? Right. So how do we do this? One of the things that we can do that was done, that was put forth in this seminal paper by Billera, Holmes, and Vogtman, is to create a space where each tree is associated with what we call an orthant, which is a subset of Rn, in particular the positive subset of Rn. And as you can see here, each axis in this figure is related to one branch. So what happens here is if I take this branch here and I collapse it to 0, then I get to this origin point. Now, if I don't have time to explain the details, but what happens in this space is it will admit geodesic. So we'll admit unique shortest paths. So if I want to go from this tree, let's say, to this tree, what I do is within the orthant, I compute the Euclidean distance up to the border. And then when I get there, I just switch over to the other side and compute the path length from the border to my destination tree. These two segments are Euclidean distances, or the equivalent norms. We usually use Euclidean norms, but it can be anything else. The point being that if I do it this way, then this will be a shortest path. It will be a geodesic. So this is what we call a cat-0 or a Hadamard space. It will have negative overall curvature, and this configuration, let's say, has implications to what sorts of things we can and can't do in tree space. But this is a useful representation that shall come back to us later. But before we get there, let's talk about MCMC. So in particular, what I'm interested in as a scientist is defining a posterior distribution over the space of trees and over the space of other things as well, other parameters of interest. And so here, D are observed data. Sometimes it's, most times it's an aligned matrix of molecular sequences, but we won't get into that today. So here, the object I'm interested in is the tree, so the graph, and then B are the lengths, the edge lengths in this graph. So both those things, when combined, will make for a phylogeny. And sometimes I'm interested, or most times I'm interested in this omega parameter, which are, for instance, the NE that we talked about a couple slides ago. So the population, the effective population size, is a parameter that somehow depends on the tree, and a parameter I'm interested in. Right. So we usually don't write the posterior distribution this way with its appropriate normalizing constant, because sometimes, or most times, we can sample from this distribution without ever having to compute this normalizing constant. And here, this is totally true as well. We're never going to have to compute this. But the reason I wrote this way here was to give you an idea of the sort of complexity involved in this problem. So in a way, I'm trying to motivate how hard the problem is by showing you how hard it would be if we had to compute this normalizing constant. So to compute this normalizing constant, we would have to integrate over omega, so integrate over a potentially high dimensional subset of the reals of Rn, really. Then after that, we would have to integrate over all of the possible configurations of branch lengths. And then when all of that was done, we would have to sum over all of the possible graph configurations, all of the possible branching orders. Now, just to give you an idea, if we had about 53 individuals in that tree, 53 leaves, we would have 10 to the 80 trees in this space. So that is about the same number of protons in the observable universe. So, astronomical doesn't quite capture the size of this space. Right, okay, so how do we actually sample from that distribution, for that target distribution that I just defined? Well, one of the things we can do is Metropolis-Hastings. So we're gonna say that tau is a phylogeny, and tau is gonna be T, the branching order, so the graph, plus the edge lengths. And so what I need is some proposal mechanism that will get me from a state tau to a state tau prime, right? So proposal mechanism and transition kernel are not the same thing, I'm sure you know, but here I will use them interchangeably, okay? Because for our purposes, it'll be fine. This is also standard knowledge. If I accept my new proposed states in the chain using this acceptance probability, then I will get a valid Markov chain. The last bit of this slide is to notice that I am making the acceptance probability somehow depend on this indexing parameter gamma. So gamma will control how bold or conservative my proposals are. So really what we're doing here is adaptive MCMC. You ought to be careful, because this adaptation needs to be done in a very careful, controlled way to ensure that the chain is stationary. So this is a very delicate thing, but it can be done. So we can adapt this chain in such a way that we can change gamma in such a way so as to achieve or approach a target acceptance probability. Right. So here's one of the things that you can do, which is what I did during my PhD. To propose a new phylogeny from a starting phylogeny. You can pick a node, any node in the tree. We're going to pick them uniformly. So after you've picked it, you find its parent and you disconnect it there. So you're disconnecting a subtree. And then what you're going to do is you're going to sample a distance delta, a patristic distance delta from a distribution. The only requirement is that it is unbounded and positive. So you're going to draw from this distribution, and then you're going to find all of the nodes in this tree that are that distance apart, right? So this is what's being shown in this panel. So the green dots are all the destination nodes that are that distance away, let's say, from the red dot, from my initial target node. And now I will again uniformly pick amongst these green um, destinations, and I'm going to reattach the parent of my red dot or my red node at the destination. And this way I have created a new phylogeny. So there are a couple of things to observe here. First, I have updated both the branch lengths, so the edge lengths, and the graph structure at the same time, right? The second thing is, this is done in such a way so as to preserve the terminal node heights. So this means we'll never propose a tree that has negative, or sorry, that has zero probability under the target. All of the new proposed states will be valid. They will have no negative probability under the target. So our proposal will never lead to an automatic rejection. Right, and the third thing is we can control how bold or conservative this proposal is by controlling the variance of this distribution. So if this distribution is in the location scale family, you can just adapt the scale of this distribution to get a particular acceptance probability. Right, sorry. Right, so these last panels are just showing you the reverse move, and it turns out that the Hastings ratios of the correction that you need in order to make sure detailed balance is achieved depends on the cardinality of this set, which in turn depends on delta. Won't get into details here. So, you know, we're dubbed this proposal subtree leap. So subtree leap will have a few properties, most of which I've already discussed. So it's adaptive, it's height constrained, it changes things simultaneously. So it proposes a new phylogeny at simultaneously. So it will change branch length and branching order. And it is a height restricted subtree prune and regraft operation for those that know what this means. So it will inherit many cool or many useful mathematical properties from this type of rearrangement. Right, so I promised in my abstract that I would tell you a little bit about the how to prove ergodicity for this sort of move. So I'll do good on my promise now. So the first thing that I told you, sort of an off remark, was that the distribution where you draw the distances needs to be positive and unbounded. I hope that will become clear now why. So the first thing is we're gonna, you know, state a theorem, but before it's useful to talk about a little remark. So the remark is subtree leap, this proposal mechanism, will induce an irreducible Markov chain on the space of graphs, so on the space of trees. So to see that that's true, you're going to go from x to y. So for any x, there is some delta depending on y, delta positive, such that the probability of going from x to y, conditional on this distance, obviously, is positive. So it's non-negative. Now, the thing is, y needs to be in the neighborhood of x. So this means that you can't go from any x to any other y arbitrarily. You need to, you can only move in the neighborhood. But the SPR graph, so the meta graph that represents this space, is connected, which means that if you have positive probability of exploring any member of your neighborhood, then you have a positive probability of having a path that gets you from anywhere to anywhere else. So this is a useful remark. By the way, the details are on my thesis if anyone is interested. So the theorem then is that this will not only lead to an irreducible Markov chain on G, but will also lead to an ergodic Markov chain on the product space of G and the appropriate subset of Rn. So this is the space of phylogenies, right? And the way to prove this is you use the remark to get you to a point where both trees have the same graph structure, only different branch lengths. And then you can use a very simple argument to establish Harris recurrence. And then you get to prove that your move is ergodic. Now, the nice thing about it being ergodic is that we can employ it as the sole update mechanism in a Markov chain, right? Usually in phylogenetics, what you do is you use a bunch of these proposal mechanisms, one of them being ergodic, so that means the whole bunch is going to lead to an ergodic Markov chain. But this guarantees that you can only use a subtree leap and that'll be fine. Now, I don't have a lot of time to tell you about many results, but this is an MDS projection. So I took a bunch of trees from a very long run, and then I computed the distance between all of them, did an MDS projection, and plotted this against posterior probability. So this should give you a representation of the target distribution. And what I'm showing you here is that subtree leap is able to explore this distribution quite well and a bit better actually than the default kernels, but this figure doesn't actually show that. Anyways, it's not the point. My point here is not to show you any compelling results, but rather to give you an idea of this whole research area. So it's much more important to me to tell you about some open problems that I think are very hot right now, very important. So there are three problems that I've arbitrarily picked. The first one is, how do we exploit the geometry of this space to make informed transitions? So in connection with, say, HMC or MALA, these techniques exploit the differential structure, so the gradient of the target, in order to make informed transitions. But our space lacks that differential structure, so we need some other sort of geometrical intuition in order to make informed transitions. The second thing is, how do we quantify exploration of the target? If I changed the distances that I used in my previous figure, the target would look completely different. So there's no canonical representation. I don't mean to say that there's no research on this area. There is, but it's very little compared to the usage of these techniques in practice. So we need more research. In particular, we need better diagnostic measures or metrics for MCMC in this space. So things like R hat or whatever, they don't work in this space. So we need other metrics. And to me, the most important open problem is the so-called optimal scaling problem. So there's a vast body of literature by people like Gareth Roberts and Jeff Rosenthal on what's the optimal acceptance probability for a large class of algorithms and a large class of target distributions. But to my knowledge, we know nothing about trees, right? And so we could approach this from a computational experimental point of view and do some experiments and see what works and what doesn't. And that's definitely something that we ought to do. But ideally, what we would like is some sort of theoretical guidance. So theoretical results to guide computational experiments. So I think that this particular problem is a tough nut to crack. And in particular, we'll crack it by combining mathematical results with computational experiments. Right, so this is a slide, I will not read it. This is a slide that shows you all of the useful statistics that's being done in the space I presented you, the BHV space. So we can do things like prove central limit theorems and do confidence sets. So, and as you will see, most of the papers and dissertations here are very recent. So this is a very hot area of research. And what we need is more mathematically minded people embarking on this train. So just to wrap up, searching trees is hard because the parameter space is complicated, it's huge, and sometimes it's, or most times it's multimodal as well, so that doesn't help things. I've showed you that height preserving rearrangement is the way to go if you're doing, you know, what I'm doing, which is estimating time-calibrated trees. So we want proposals that don't propose things that are impossible under the target. We really ought to be moving in the direction of adaptive moves, so things that can control the variance of the proposal so that you achieve a certain acceptance probability. Otherwise, when the tree size grows, we start making horrible proposals, so really low probability proposals, and that's not what we want. And the last bit, which is the most important, is there's loads and loads more work to be done to bridge the gap between the data that we have and the models that we want to fit. Right now, we have a lot of data, and we have some models that we would like to fit to data, but these things just don't match. So we end up fitting very simple models, and even then, that takes days or sometimes weeks to fit. So we ought to correct that as soon as possible. So thank you for your attention. I'll stop talking now.